Welcome to development of a cell-based reporter assay for measuring ADAR1 editing activity for drug discovery and cell biology research, presented by BioCompare and sponsored by BPS Bioscience. My name is Tamlin Oliver, a managing editor of BioCompare, and I'll be the moderator for this discussion. Today we'll be joined by Valerie Sapp, a research scientist at BPS Bioscience, and Veronique Barron, Associate Director, Scientific Applications at BPS. Our speakers will talk about the high-throughput cell-based assay for measuring ADAR1 editing activity that BPS Bioscience has developed and validated. In their talks, they'll give an overview of RNA editing, discuss the function of ADAR1, and then describe the principle of these new cell-based assays, present data, and discuss the relevant applications of the three cell lines. Before we get started, though, I want to quickly go over a few procedural details. We'll hold a live Q&A session at the end of the discussion. You can submit a question for the panel at any time using the Ask a Question tab in the upper right corner of your screen. The right side also features an overview of the webinar as well as the speaker's bios. If you have any technical difficulties at any time, click on the Test Your Connection button. From there, you can access webinar support. We also have social media widgets beneath the webinar window to facilitate the sharing of this webinar with colleagues. All right, with that done, I can properly introduce our panelists and we can get this webinar started. Valerie is a cellular biologist with research expertise and publication record in stem cell biology, CRISPR screening, and cellular assay development. Dr. Sapp received her PhD in biomedical sciences from UC San Diego in 2020 and has been a research scientist in cell-based assay development at BPS Bioscience for the last 3 years. Veronique is a cellular and molecular biologist with extensive research experience in drug discovery and development and tumor cell biology. Dr. Barron has worked in a variety of research environments, including governmental and not-for-profit institutions, biotech, and pharmaceutical companies. She's published numerous patents and scientific manuscripts, and for many years sat on grant review panels for the DoD. Dr. Barron has been a scientific applications manager at BPS for the last 3 years. We're ready to get started. Valerie, you're up first.
Thank you, Tamlin, for the kind introduction. It's a pleasure to be able to speak with you all today about the exciting field of ADAR biology and our work here at BPS Bioscience developing validated cell-based assays for ADAR1 activity. To give you a brief overview of our presentation objectives for today, I will begin by introduction to ADAR1 editing and RNA editing in general. I will give an overview of our ADAR1 reporter cell line development process and an overview of our ADAR1 reporter cell line applications. And we'll finish off with a discussion of our ongoing work and future directions. To help you better understand our ADAR1 product offerings, I will give a brief introduction to the biological role of RNA editing, an introduction to the ADAR family of enzymes, the role of ADAR1 in health and disease, and the applications of ADAR technologies. So what is RNA editing? RNA editing refers to the irreversible conversion of one RNA base to another. This is distinct from other RNA modifications that are more epitranscriptomic, such as RNA methylation. And RNA editing plays a biological role in a variety of processes. RNA editing has several different outcomes, and these can result in transcriptomic diversity, when the RNA edit switches one RNA base to another, and this results in a change in the amino acid codon. In the coding variation, this can result in a protein that is different from what is encoded in the genome. Additionally, if the RNA edit occurs at a splice junction, this can introduce different alternative splice variants. RNA editing also is involved in modulating the activity of microRNAs, and this can change the expression levels of the target protein of interest. And additionally, and most relevant for ADAR1, is the regulation of the immune system through the sensing of double-stranded RNA, also known as the recognition of self. There are two main families of RNA editors, ADAR and APOBEC. The ADAR enzymes convert adenosine to inosine through the process of deamination, while APOBECs convert cytosine to uracil. And ADAR edits primarily occur within double-stranded RNA, and APOBEC edits primarily occur within single-stranded RNA. To give you a brief introduction to the ADAR family of enzymes, it's comprised of ADARs 1, 2, and 3. While ADAR3 is catalytically inactive, ADAR1 and 2 have substantial overlap in their sequence as well as their structure, and this structural similarity leads to an overlap in the target sequences of RNA editing for ADAR1 and ADAR2. The ADAR1 is encoded by two primary protein isoforms, the P150 and P110 isoforms. The P150 isoform, which is longer, also includes a nuclear exit sequence. This can result in the cytosolic presence of the ADAR P150 isoform compared to the nuclear presence of the ADAR1 P110 isoform. Additionally, in this P150 isoform, there is the Z-alpha binding domain, which is responsible for the binding to Z-DNA and can mediate editing-independent functions of ADAR1. ADAR1 plays a biological role in the regulation of the immune system through modulating the immune activating properties of double-stranded RNAs. So double-stranded RNAs are an immune activating signal, danger signal that are sensed by double-stranded RNA sensing proteins. ADAR1 exerts its role by converting adenosines to inosines within this double-stranded RNA, leading to the lack of recognition by downstream double-stranded RNA sensors. Some endogenous RNAs, such as repetitive elements, also display the double-stranded structure, and this is important for the role of ADAR1 in modulating the lack of response to those self-double-stranded RNAs. The ADAR P150 isoform drives the resolution of inflammation due to its cytosolic expression and inactivation of viral double-stranded RNAs, and loss-of-function mutations in ADAR1 result in autoimmune diseases. ADAR1 is an interesting therapy for cancer targeting. ADAR1 has some direct effects in tumor biology, including having many tumor suppressor genes and oncogenes that are ADAR1 editing targets, and ADAR1 gene duplications are observed in several cancers. More recently, a role has been recognized for the intersection of ADAR1 and tumor immunology. ADAR1 regulates the response to tumor-derived interferons by dampening the innate immune response. It can also intersect with immune checkpoint inhibition, where ADAR1 knockout enhances the response to checkpoint inhibitors such as PD-1 and PD-L1 blockade. So what are the applications of ADAR technologies? ADAR technologies can be used to characterize different ADAR1 isoforms and mutants, to screen for different ADAR1 modulators, such as small molecule inhibitors, RNAi, and antisense oligonucleotides, and as mentioned before, is relevant for cancer immunotherapy targeting. Additionally, there are several companies that are screening for inhibitors of the Z-DNA binding domain of ADAR1, which would modulate its interaction with the Z-DNA and influence both its RNA editing function in the DNA as well as the RNA-independent functions of ADAR1. Additionally, ADAR1 is being used to develop programmable RNA editing and RNA sensing systems. The RNA editing systems are similar to what is being developed with Cas9 for DNA, where you would be able to swap the base at a specific location, guided by a guide RNA and directed to your specific location in the RNA and change the protein that is coded. Now that we have an idea of the role of ADAR in biology and disease, what are the relevant applications here for characterizing ADAR1 activity? ADAR1 activity is typically characterized by a few different methods, including RNA sequencing, next-gen sequencing or transcriptomic sequencing to characterize the entire ADAR editome, as well as targeted sequencing of specific regions and transcripts. Additionally, there are many biochemical assays available for identifying ADAR1 binding and editing inhibitors and modulators, including here at BPS Bioscience, our ADAR1 binding assays. And finally, there are cell-based editing reporter assays for understanding ADAR1 activity, and that's what we'll be speaking about today. So today I will walk you through the design rationale of our cell-based reporter assays, our reporter screening and selection process, and our cell line development process. As we discuss the design of this reporter system, it's important to note that this design does differ substantially from many of the reporter systems that are out there. Typically, when we think about a luciferase reporter system, we think about the reporter firefly luciferase being encoded downstream of a particular promoter or response element that's downstream of your pathway of interest. in the case of this particular reporter design, we're doing something a little bit different. This ADAR1 reporter system is constitutively expressed, and the firefly luciferase reporter is only expressed when ADAR1 is active. Let me further explain. So our reporter system here encodes a editing susceptible hairpin with a stop codon that is converted when ADAR1 is active to a tryptophan codon. So what this means is without the presence of ADAR1, we have a stop codon present and no downstream expression of firefly luciferase. When ADAR1 is present, the stop codon is converted to a tryptophan, and we do have a resulting downstream expression of firefly luciferase. Now that we have an understanding of the reporter system that we're working with, we will be performing all of these experiments in HEC293 cells, which have low constitutive ADAR expression. To select our specific reporter sequence for our ADAR1 cell-based assay, we designed and constructed 3 alternative hairpins and generated their respective cell pools. These cell pools were tested for their luciferase response to transient transfection of ADAR1. And so what we can see here is that we start out with a low baseline signal for the firefly luciferase, and upon expression of increasing concentrations of ADAR1 plasmid, we see an increase in the firefly luciferase activity. This is true for both reporters 1 and 2, while the third reporter that we tested was inactive. Since the therapeutic target of interest for a drug discovery field is ADAR1, we were interested in designing an ADAR1 biased reporter. To assess this, we tested the response of reporters 1 and 2 to both ADAR1 and ADAR2. And what we saw is that both of these reporters have a substantial response to ADAR1, and for ADAR2, they have a more minimal response. And while we do see some increase in signal from reporter number 1, we see no increase in signal for reporter number 2 with increasing concentrations of ADAR2 plasmid. To this end, we have identified an ADAR1 biased reporter for reporter number 2. We then leveraged this ADAR1 biased reporter to generate the first cell line in our portfolio, which is our ADAR1 responsive luciferase reporter cell line. We further confirmed in this clonal cell line that it responds well to stimulation with the ADAR1 P150 isoform, and that we retain a minimal expression of firefly luciferase in response to ADAR2 expression. Additionally, we tested the response to the ADAR P110 isoform, and we found that we do see some signal activation with the expression of ADAR P110 isoform, although it is less than the ADAR P150 isoform. And we believe that this is in response to the different localization of ADAR1 P150 isoform versus ADAR1 P110 isoform. I'd like to review the applications of this first cell line, our ADAR1 responsive luciferase reporter cell line. This cell line is useful for those customers who are interested in screening for different ADAR1 isoforms, mutants, or variants activities in RNA editing. As ADAR1 is an interesting target for cancer immunotherapy, we next went on to generate a cell line that constitutively expresses ADAR1 to facilitate the screening for modulators of ADAR1 activity. So this second cell line in our portfolio is our ADAR1 activity luciferase reporter cell line, which, as I mentioned, we've constitutively overexpressed ADAR1. This is confirmed via Western blot. And because of the constitutive expression of ADAR1, we see here that we have a constitutive activity for the luciferase reporter. We then further validated this cell line through siRNA treatment and knockdown of ADAR1 expression. And what we see is when we knock down ADAR1 expression, we have a decrease in that luciferase reporter signal, indicating that we are, in fact, detecting bona fide ADAR1 activity in this cell line. We did further validation to ensure that this is not due to a decrease in cell viability. And what we can see here on the right is that we have no decrease in cell viability with our siRNA observed. So this signal is coming entirely from a decrease in ADAR1 expression. We next went on to validate the signal window of our ADAR1 activity luciferase reporter cell line. And we did this by comparing the firefly luciferase of this ADAR1 overexpressing reporter cell line to the parental reporter cell line, which does not overexpress ADAR1. And what we see is that we have a sufficient signal window in both a 96-well and 384-well plate format to detect modulation and decrease in ADAR1 signal in this ADAR1 activity reporter cell line that we have. We found that the best conditions for this difference in the signal window is in the 96-well format, where we have excluded the edge wells. The relevant applications of our ADAR1 activity luciferase reporter cell line is to screen for small molecule modulators or RNAi antisense oligonucleotides against ADAR1. Many of the commercially available ADAR1 inhibitors exhibit substantial cytotoxicity, making counter screens for cell viability necessary when evaluating ADAR1 inhibitors. In order to address this, we've generated a third cell line in our portfolio based upon our second cell line in the portfolio, and this also overexpresses renilloluciferase constitutively. And this renilloluciferase is going to be our counter screen control for cell viability. This cell line is compatible with our two-step luciferase reagent. And what we have here is at cell densities that are relevant for cytotoxicity assessment, that is those cell densities that are at or below the plating density for the assay, we see that both the ADAR1 firefly luciferase reporter signal and the renilloluciferase signal for cell viability track well with one another. And at these relevant plating densities, this firefly to renilloluciferase ratio is equivalent. We further assessed this cell line by evaluating existing commercially available ADAR1 inhibitor, fludarabine. And when we assess fludarabine in this system, what we observe is that we see a decrease in both the ADAR1 responsive firefly luciferase signal here in blue and the constitutive renilloluciferase signal here in green. But we see that the firefly luciferase signal from ADAR1 activity is decreasing faster or more than the constitutive renilloluciferase signal. And what that means is that we're seeing more of a decrease in the ADAR1 activity than we are seeing a decrease in cell viability. And so when we perform an analysis of the firefly ADAR1 activity luciferase ratio to the renilla cell viability ratio, we see that we have a modest decrease in this metric with increasing concentrations of the inhibitor fludarabine. In the future, we plan to validate more ADAR1 inhibitors as they become commercially available. Similar to our second ADAR1 activity luciferase reporter cell line, this cell line is relevant for screening for small molecules or RNAi antisense oligonucleotides that modulate ADAR1 activity. And this one has the added benefit of the ability to simultaneously assess these modulators for their toxicity via the renilloluciferase signal that we have. So I'll give you here a recap of the applications of our ADAR1 luciferase reporter cell line products and give you some insight into choosing the correct cell lines for your needs. So the first cell line in the portfolio that I described was our ADAR1 responsive luciferase reporter. As mentioned before, this is useful for screening the activity of different ADAR1 isoforms, mutants, or variants that may be of interest to you. And it's useful as a baseline control, as described for our ADAR1 activity reporters. This allows you to characterize and validate the signal window of this reporter and ensure that you have a good assay window to screen for your inhibitors. Our second cell line, the ADAR1 activity luciferase reporter cell line, which constitutively expresses low ADAR1 and has exogenous high overexpression of ADAR1. It's useful for screening modulators of ADAR1 activity. And our third cell line, which is the 2-luciferase reporter cell line, is useful for screening for these modulators of ADAR1 activity. And additionally, because of the expression of the renilloluciferase, it has a good simultaneous readout for ADAR1 modulators toxicity. As a brief overview, we discussed ADAR1, which is an exciting target for drug discovery and development for cancer and immunotherapy. Here at BPS Bioscience, we've generated 3 ADAR1 reporter cell lines to meet your research needs. And for the future directions, it's up to you. You may be interested in performing isoform screening with our ADAR1 responsive reporter HEC293 cell line, or performing inhibitor screening in our ADAR1 activity reporter HEC293 cell lines, both of which we can do here for you at BPS Bioscience as a service. Additionally, you may be interested in introducing our ADAR1 reporter system into your cell line of interest, which we can perform as a custom cell line development project. Thank you so much for your attention. We look forward to answering your questions at the end. And I'll hand it off to Veronique now. Thank you.
Well, thank you, Valerie. It was a terrific presentation of the cell lines that you have developed for BPS Bioscience. Now, BPS Bioscience offers other related products to support your research needs. And since Valerie talked about measuring luciferase activity in a reporter cells using the one-step or the two-step assay systems, I will start with these. In the one-step system, two components are provided. They are mixed together right before use, and the single reagent is added to the cells and performs the cell lysis while adding the luciferin substrate in one step. It is not necessary to wash the cells either before or after. You just add the reagent onto the medium and you read the results 15 minutes later. In the two-step assay system, on the other hand, you multiplex the measurements of firefly and granular luciferase activities. With a two-step, you prepare two reagents. The first corresponds to the west step, which performs cell lysis and monitors the activity of firefly luciferase. After the firefly reading, the second reagent is added directly to that plate to monitor the activity of granular luciferase. Again, you just add the reagent and read the results. So next, I would like to talk about an assay kit that measures ADAR1 binding to an RNA probe, and that's the ADAR1 RNA TRFRET assay kit, in which TRFRET, of course, stands for Time Resolved Fluorescence Resonance Energy Transfer. This assay measures the binding of ADAR1 to an RNA probe. First, the recombinant purified ADAR1, which contains a flag tag, is incubated with a biotinylated RNA probe to form a complex. Streptavidin, conjugated to an acceptor, binds to the biotin on the RNA probe, and an anti-flag antibody, conjugated to a terbium donor, binds to the flag-tagged ADAR1. Upon excitation, the fluorescence donor emits energy, which excites the acceptor, which then fluoresces in the red. And this happens when the complex has been formed and the acceptor and the donor pair are in proximity. So this assay is useful to analyze RNA binding kinetics or to study the competitive effect of ADAR1-directed compounds, such as, for example, aptamers, or small molecules that would bind within the RNA binding pocket and compete with the RNA probe. The assay proper is very simple. Having no wash steps, it's also very fast. The protein is pre-incubated with a compound of interest for 30 minutes, and a biotinylated probe is added for 15 minutes before the FRET donor and acceptor are added for another 30 minutes. And then you just read the results in a TRFRET capable instrument. So on the next slide, I am showing example results obtained using this assay. So the top graph shows that the FRET signal is directly proportional to the binding of ADAR1 to RNA. As you can see, it increases when you increase the amount of ADAR1 that is added to the assay. And on the bottom graph, you see an experiment in which increasing concentrations of a competitor RNA were incubated with ADAR1. And as you can see, the IC50 of the competitor can be determined with precision. And with this, I will end my presentation. I'll pass it over to Tamlin and take any questions you may have.
Before we start the Q&A, I just want to point out that we have some additional resources on ADAR1 from BPS Bioscience in the resource tab, as well as a certificate of attendance that you can download. Also want to let you know that we have a bunch of questions already to get started, but we have plenty of time to answer any questions you might have. So I encourage you to please submit your questions. Veronique is here and she can answer them for you. So, all right, Veronique, are you ready?
I'm ready. Hi.
Okay, let's get started. The first question, did the transfection of ADAR1 in HEK293 cells to generate the ADAR activity cells affect cell growth or health?
Oh, that's a good question. So no, it did not. Overexpression of ADAR1 in the HK cells, as far as we can tell, did not affect cell growth. It did not affect the health of the cells, and it didn't induce any toxicity to the cells. The only thing that we did observe is that in the two luciferase cells, which have the two constructs for firefly and for Renilla, these cells grow just a touch slower, but that's probably because of the selection antibiotics. They have more selection antibiotics than the simple transfected cells, but ADAR itself did not affect the growth of the cells.
Okay, the next question, what is the size of the hairpin that was introduced into the cells as ADAR reporter?
The hairpin itself is quite small, so it's about 20 to 30 base pairs. The construct is a little larger, obviously, because it contains the sequence encoding the firefly luciferase. Yeah, the hairpin is fairly small compared to the construct itself, so about 20, 30 base pairs.
All right, I have a multi-part question here next. Question about the assay kit. Is the enzyme enzymatically active? If yes, should the user expect a drop of signal after a long incubation due to RNA probe release based on dynamic kiss and run mechanism?
Right. So the protein that we use for the assay is the full-length protein. It is expected to be active. However, BPS Bioscience does not have an activity assay for the protein. We know that scientists who have used our protein have described activity in their own publications. So if you were to go on our website and look in the citation tab for the protein itself, you would see that there are two publications showing the protein has activity. The assay, however, does not look at the activity of the protein. It only looks at the RNA binding. What we see, I think that we have tested an incubation of the RNA with ADAR1 for up to 2 hours, and we do not see any drop in signal. So we don't think that there is this mechanism where it would change the RNA and release it. We haven't observed that in our assay.
Okay, before we go on to the next question, I just want to remind everyone that we can still probably have time to answer your questions, so please do submit them. All right, next question. Have you tested any other commercially available ADAR1 inhibitors in your cell lines?
So yes, we have. If I remember correctly, we've tested the 8 adenosine, I think, which is not an ADAR1 inhibitor proper. However, it has been described to decrease the activity of ADAR1. It's not as efficient as fludarabine when we use it in the cells. It's just that there's no real ADAR1 inhibitor that is commercially available that we could test. Fludarabine is the best one that we have found. And all these inhibitors were initially described for other targets. They're weak inhibitors of ADAR1 and they have some toxicity. So for example, the problem we have with the 8 adenosine is that it's also toxic to the cells, and it's hard to make the difference between the decrease in signal that is due to the death of the cells and the decrease of signal that is due to the inhibition of ADAR1. So when we use the two luciferase system, we can make out these two effects. So we can make out the toxicity of the inhibitor and we can make out the activity of ADAR1. So we see that The 5-ASA has quite a bit of toxicity, and the fludarabine is actually a better inhibitor. That still induces toxicity, but has a better inhibition of ADAR1 activity.
Okay, thank you for answering those questions. We don't have any more questions coming in, so I'm going to wrap this up unless something comes in the next couple of seconds. All right, just a reminder about those resources. Check out those resources in the resource tab, and also get your certificate of attendance if you want that. And oh, here we go. I'm glad I waited. All right, we have another question. Can any of your assays be used to correlate binding activity with RNA-seq-based detection to validate if a specific bound RNA is edited?
So I think you could use RNA-seq-based detection in the cell lines. I don't actually know if we have tried that ourselves. I'm not sure. Let me see. So yes, it could be used in the cell lines. We haven't really tried, and we have to keep in mind in that case that if you're using RNA-seq, so you are going to measure the effect of ADAR1 on the construct itself. Now, let's remember that we also have several proteins that we may make from the transcript, and then we measure the enzymatic activity of these proteins. So the signal is amplified. So compared to an RNA-seq sequence, we'll see more effect on the luciferase activity than we would see on the construct itself because of that little amplification effect. But basically, yes, you could use RNA-seq to validate the editing of the RNA.
Okay, thanks, Veronique, for tackling that question. All right, we don't have any more questions, so I am going to wrap this up. I want to thank Valerie and Veronique for joining us today to share their expertise, and give a special thank you to BPS Bioscience for sponsoring today's event. This webinar will be available on demand in about 24 hours. If you registered, you'll get an email from us confirming that. You can watch it again, share it with colleagues, or just check out those resources. Also, if you go to biocompare.com, you can see our upcoming webinars. We have a CAR T-cell webinar coming up, and see our archives. We have tons of webinars we've done over the last 2 years. So Veronique, thank you so much. And that's it for me. We'll wrap this up. Thanks for joining us today, and
Thank you.
we look forward to seeing you at our next event.
Thank you very much. Goodbye.
You're welcome.